Welcome to Equine Guelph's Research Radio, a monthly look at cutting-edge research being done at Equine Guelph, brought to you by SSG Gloves. Our guest is Dr. Scott Weese, talking about the latest in gut function research. Dr. Weese joins us in a moment, but first, SSG is on the cutting edge of premier gloves. The original SSG all-weather glove is our most popular. Quality, durability, fit, and comfort perfect for any equine discipline with its AquaSuede Plus palm and elasticized back for cool, comfortable fit. All you have to do is throw it in the wash and let it air dry. The original, the SSG all-weather glove. See your SSG dealer for details. Dr. Reese, welcome to Research Radio.
Yeah, thanks for having me.
You know, gut function is something we lay people find fascinating because most of us aren't aware that it affects so many aspects of equine health. Tell us about that.
Well, we're just actually starting to figure out some of these aspects, and it's not just horses, it's people who are getting a lot of this information. In the past, we've known the gut's important for colic and diarrhea and other things, but as we learn more about horses and other species, we realize that the gut really plays a key role in a wide range of diseases that go beyond the gut.
Now tell us about those.
Well, we're still just trying to sort this out in horses, and one of the problems we've had in the past is the horse gut is so big and so complex, we really haven't been able to figure it out. And we know now that we've got thousands of different bacterial species that live in the horse's gut, and they change throughout different parts of the horse's intestinal tract. And they do some very strange and complex interactions that we're only now starting to wrap our heads around. In the past, what we could do is take a fecal sample, a manure sample, and put it on a culture plate. We'd only grow a few different bacteria, and probably not most of the ones that are important because they just don't grow well in the lab. So now that we can do things with DNA sequencing, we can take a sample and we can pull out millions of DNA sequences that represent different bacteria and figure out these thousands of bacteria that are present in the gut. And that's letting us start to get a bit of a snapshot of what the normal gut's like and how that changes in certain types of diseases.
How does gut function affect things like colic and more importantly, laminitis, I think?
Well, again, we're still trying to sort this out, but the colic is fairly straightforward for some types. Now, one of the problems with colic is colic's not the same thing. There are a whole range of them, but a lot of colics will relate to gas production and changes in motility of the intestinal tract. And gas is produced by bacteria when they ferment different things. So if you get changes in those bacterial populations, you can get more gas production that could cause colic itself or might predispose the gut to going into other locations, to twisting or displacing. So that's one aspect I think we can more easily talk about, the gut microbiota and colic. But there are other things that happen too. The gut microbiota probably interacts with the immune system, with the wall of the intestine that actually controls motility. So it could be that the bacteria in the gut alter how that gut works. And sometimes that's good, and sometimes that's bad. And if it slows down how the gut's moving, that can allow gas to build up or manure to build up and cause an impaction. So with laminitis, it's a little more complex because we're looking at something that's happening outside the gut, but which the gut relates to. And this is something we've known for a while, that certain changes in the intestinal tract can predispose by laminitis. So you get changes in the bacterial populations in the intestinal tract, and these bacteria produce all kinds of byproducts. And some of these are good for the horse, and some of these are bad, and some are toxins. And when you get some changes in the bacterial population, you get production of these toxins that can then go in the horse's circulation and cause inflammation, and in some cases cause laminitis. So a gut change, changing what's going on in a distant body site. Now, laminitis is fairly well understood, and we know that if you take a horse and you give it a lot of grain, or you put certain horses out in a really lush pasture, they're prone to laminitis, and that's because of changes that occur in their gut.
More with the good doctor in just a moment. But here's a reminder from SSG Gloves. Go with the glove used by champions. Champions like Eric LeMay's. The SSG Digital is the ultimate in feel, grip, breathability, and durability. The digital pattern increases grip and wear, and it's machine washable. See your SSG dealer for details. Now, you're using DNA to more clearly define bacterial problems. Tell us how that works.
Well, what we do is we take advantage of what we can do with sequencing now. As opposed to trying to grow things, we just look at their DNA. Because they don't have to be alive. They don't have to be something we can grow. They don't even have to be something that we know, because we can identify an organism without actually knowing what its name is. So we can take a manure sample or a sample of anything else that might have bacteria in it, and we try to amplify one part of that bacterium. And then we look at the DNA sequence, and that tells us what that bacterium is and what it's related to. And with that, we can start to infer what it's doing, why it's changing, how it's changing, and what those changes might represent to the horse.
How does that work from a preventative medical standpoint?
Well, at this point, it's hard to extrapolate it right to the horse because we're still very early in learning. So we can't take a sample from a horse and look at it and say, okay, your horse is healthy or not healthy. But we're starting to get information that's going to help us. And as we do some new studies, we're getting to the point where we might be able to use the gut kind of signature as an indication of health. In one recent study we did, we were looking at mares that were colicking around the time of giving birth. And we got manure samples from these mares before they foaled and after they foaled. Because around the time of foaling is a time that's high risk for colic. And within that group, we got some mares that we had samples before they colicked, and we compared them to samples from mares that didn't colic. And we saw changes there. So we saw changes that were occurring in their manure about a week before they actually developed signs of colic. So this is kind of suggesting that we might be able to see these little signs that occur before a horse gets sick. And that's really the key. If we can figure out what's occurring before the horse gets sick, we might be able to detect that and intervene. It also lets us figure out why colic happens, as opposed to looking at what's happening at the time the horse is sick, which doesn't tell us necessarily as much, looking at what happens in the time that leads up to the horse getting sick. So things are changing fairly rapidly, and we're hoping in the future we'll be able to say, yes, this is a horse that's got something going on that's predisposing it to colic, or yes, we understand this change causes colic, so let's do things to make sure that change doesn't happen.
Ounce of prevention.
Prevention is a big thing, yeah, absolutely.
And that's where this falls into play. Well, Dr. Weese, as always, a pleasure having you on Research Radio. We thank you for your time, and best of luck in your continued research.
Thanks, Norm. Have a good day.